Hey guys, in this video we'll be talking about the models of DNA replication. There are three proposed models for DNA replication, and the first model is the conservative model. This model proposed that the parent molecule divides itself into four strands. The first two are the original strands, while the next two are the new daughter strands. The daughter strands form a newly synthesized molecule. Meanwhile, the two original strands reform the parent molecule. The next type of model is the semi-conservative model. Although this model was founded by Watson and Crick, it was experimentally proved and verified by Meselson and Stahl. This model proposed that each new molecule of DNA would contain one strand of the original complementary DNA molecule and one new parent strand. The original complementary DNA molecule is the one in green, and the new parent strand is the one in red. The next model proposed by Max Delbruck is the dispersive model. This model proposed that the parental DNA molecules were broken into fragments and that both the strands of DNA in each of the daughter molecules were made up of an assortment of parental and new DNA. Matthew Meselson and Franklin Stahl conducted an experiment and concluded that DNA replication is semi-conservative. They used isotopes of nitrogen in the experiment. This is because nitrogen is an essential component of DNA and because nitrogen has both heavy and light forms, which could bring about distinguishing factors. In the first part of the experiment, they grew bacteria in a liquid culture medium containing 15N. 15N is the heavier form of nitrogen. This is why the single band formed was at the bottom of the tube, because of its high density. In the second part of the experiment, they transferred the sample into the 14N medium and allowed for one round of replication. They then spun it and they found out that a single band in the middle of the tube, which was the intermediate between the 14N and the 15N, was formed. In the third part of the experiment, they allowed for a second round of replication, after which they spun the tube. They found out that the intermediate band remained, but in addition to that was the formation of another band higher up the tube that contained only 14N. After a second round of replication in the 14N medium, the DNA appeared as two bands, one in the position of the hybrid DNA, as seen in the previous experiment, and the other in the position of DNA that contained only 14N, which means that this position was the highest. Recall that in the first part of the experiment, there was only a single band of dense 15N at the bottom of the tube. This was the parent molecule of the DNA. In the second part, an intermediate band was formed, which means that it contained part of the 15N, which is the parent strand, and part of the 14N, which is the newly synthesized daughter strand. In the third part of the experiment, after the second round of replication, the intermediate strand still retained itself and a less dense 14N band formed. In the fourth part of the experiment, they repeated the same procedure and found out that the results remained the same.